Today we will focus on best practices of the RNA-Seq workflows. And to set the expectation, this is meant to be a very high-level overview. Experienced users looking for more in-depth information are welcome to contact tech support for project-specific questions. So today we will look at understanding the workflow, learning how to determine a successful library prep, and understanding the best practices. So let's start with the best practices. In part one, we touched upon the different kit options available in RNA workflows. You may remember this matrix from part one, which provides a nice comparison of the kits discussed. I have provided it here as a review. Now, once we complete our selected workflow, how will we know what a successful library prep looks like? We have validated our workflows with the use of bioanalyzer or fragment analyzer. However, if you do not have access to these specific instruments, something similar, such as a tape station trace, will also work. Here we have an example of a fragment analyzer trace on the upper right and a bioanalyzer trace on the lower left. And we can see that the idea is similar in that they have an upper and a lower size marker, and they are essentially showing the amount of fragments that we have at the various base pair sizes. So for example, in the bioanalyzer trace, we see that we have more fragments at the 261 base pair size than we do at the 400 base pair size. So this trace is very useful in telling us what our sample or pool looks like in terms of size composition. So if we run our RNA samples on a bioanalyzer or a fragment analyzer, this is what we would expect to see. So we have the lower marker at around 15 base pairs, and the upper marker is at about 1,500 base pairs. And this might vary depending on what chip you're using. If we're following the standard fragmentation protocol, we expect to see a smooth bell-shaped curve with a peak around 260 to 300 base pairs. Depending on what your experimental question is, there are options to adjust your fragmentation time, and this will extend the fragment size, and it'll give you a little bit of a longer read length. Adjusting fragmentation times will shift the library peak, right? So the size will change. Smaller fragments will shift the peak to the left, and larger fragments will shift the peak to the right. And what we're looking for here are any peaks outside of your main or your 260 base pair bell-shaped peak. Now, I'm not going to get into too much detail on the traces, as more detail is covered in our library QC and troubleshooting with the bioanalyzer webinar. So keep an eye out for that. I have also provided the registration link for this webinar in our resources slide. If you happen to see any additional peaks in your trace, you can always email in your trace to Library Prep Tech Support at techsupport@illumina.com. We would be happy to take a look, see if anything is atypical, and provide some feedback. Now let's review our library prep best practices for the following areas. We can look at input, fragmentation, handling magnetic beads, the equipment needed, other best practices, and library QC and quantification. Let's start with RNA integrity. When we're talking about RNA, one thing we like to focus on is the RNA integrity or RNA quality. For these kits, we refer to RNA integrity in terms of RIN or RIN number, which stands for RNA integrity number. You may also hear it also referred to as RQN sometimes, RNA quality number. It's the same thing. And what this is, it's the ratio of the 18S and the 28S rRNA peaks. And this number gives us an idea of the quality of RNA and how intact it is. RIN values are specific for mammalian samples. So if you are using a species other than mammalian, I recommend checking the literature to see how they assess RNA integrity. So looking at this diagram, the graph on the top would be an example of a sample with a RIN value of 10. We see a nice sharp peak indicating intact RNA for the 18S and the 28S regions. Once degraded, for example, let's say FFPE or samples that have been stored for a long time, we will still see a peak at 18 and 28S, but it's not as strong as exemplified by the second trace, in which we have a RIN value of 5. You'll also notice that due to degradation, we are getting some fragments in the smaller base pair regions. Now, really strongly degraded samples are shown in the lower panel. And you might notice here that you can't make out the 18S and the 28S peaks quite as well. And there are a lot of smaller sized fragments. So this would be indicative of strongly degraded material. For TruSeq RNA v2 and the stranded mRNA kits, we want the RIN to be equal to or greater than 8. And part of the reason for this is because of how we select the transcripts. We are selecting off of the 3' poly A tail. So if the RNA is fragmented, we will lose the 5' end of the transcript and maybe even the middle. You can now understand that if we use a sample of lower RIN value, how we may end up with 3' bias, low or no library yield, or incorrect library size due to smaller fragments being lost during the wash. We may also see a library that appears small, or observe unexpected peaks, such as adapter dimers, which can form when there's not enough input present. It's also possible to have high % mapping to contaminants. And when I say contaminants, I mean that we are mapping to different species of RNA that we didn't mean to capture. Having other species of RNA will cause nonspecific alignment to the reference genome and higher alignment to the contaminating RNA species, thus reducing the % alignment. High % duplicates can result when contaminants are present, as they can amplify over and over again during the PCR step. If using stranded total RNA kits, it is okay to use degraded material with a modified fragmentation time depending on how degraded our input is. Recommendations on how to modify fragment time can be found in the TruSeq Stranded Total RNA User Guide. To keep the RNA integrity, we want to avoid extended pauses until the RNA is converted to double-stranded DNA. Avoid multiple freeze thaws of input RNA. Store the RNA itself in RNase-free water or TE buffer. And this can, depending on the method, be used for up to a year, depending on that extraction method. We want to keep thawed RNA and reagents on ice and ensure the thermocycler lid is preheated for each synthesis step. Now next, we want to take a look at the input quantification. For input quantification, we want to use the recommended total input required for the workflow that we have selected. There is an option to start with previously purified RNA. So if you have your own method of how you would like to purify that RNA, you can start with your purified input, which is typically less than the amount of total RNA required. We do recommend that you use a fluorometric-based method and not a UV-based method, such as nanodrop. And the reason is that UV methods tend to be inconsistent. We also recommend a DNase treatment. Most RNA extraction kits include a method of DNase treatment, so it shouldn't be too difficult to include this step. So what can happen if we don't accurately quantify? We can get the same results as previously described in addition to reduced percent strandedness if you're using a stranded kit. Now let's look at these upfront steps. In stranded mRNA, we do a poly-A selection, and in stranded total methods, we do a ribo-depletion method. If using poly-A or Ribo-Zero, and note for right now I'm referring to the legacy Ribo-Zero beads, we want to ensure that we do not freeze these beads. Once frozen, they lose their ability to bind to magnets and reagents, and they're no longer usable. For our new Ribo-Zero plus reagent, with this method, it does not use beads, but rather it's an enzymatic depletion. So for this, we want to ensure that the consumables where we are using beads, such as the RNA Clean XP beads, that they've been at room temperature for at least 30 minutes. Cold beads do not function as efficiently, so this is an important step. We also want to ensure that the thermocycler lid is preheated and that the thermocycler must be capable of ramping at 0.1 degrees per second for the best results. So whether it's an Illumina bead or a consumable bead, we definitely want to get it up to room temperature and not have those beads frozen. In regards to fragmentation, for intact RNA, I suggest that you use the recommendations provided in the protocol. This will give us the best and most uniform coverage across the gene. You can alter the fragmentation time, but by doing so, you can get reduced coverage across the transcript. If using degraded material, such as with stranded total RNA, keep in mind that we don't want to over-shorten our libraries, hence the adjustment in fragmentation time for TruSeq Stranded Total RNA, because we don't want to obtain various insert length sizes. Possible consequences of altered fragmentation considerations is no or low library yield and less uniform coverage across the genes. And this is something more or less important depending on your experimental design. So if you are doing differential expression, you may not need even coverage. But if you're doing transcript assembly, then you may want to stick with the recommended fragmentation time to get that even coverage across the genes. Modification of fragment time is only applicable to the TruSeq Stranded Total RNA and not to the Illumina Stranded Total RNA kit. Now, our Illumina Stranded mRNA and Total RNA kits utilize anchors for adapter ligation. When using these kits, keep in mind that the RNA index anchor plates are green, and all wells are single-use only. This will be separate from the index plate, so that's a whole different plate. Add the reagents in the order listed, and add the stop ligation reagent with STL buffer before the cleanup step. If these best practices are not performed, it can result in inefficient addition of adapters or libraries that are not properly formed, resulting in low yield. Now on to handling magnetic beads. We typically use Ampere bead cleanups at the end of the reaction because they allow us to size select. Now, based on the ratio of bead solution to sample, we can select a fragment size to retain for the next step. With the first bead cleanup, the larger nucleic acids are preferentially maintained, and we get rid of the smaller nucleic acids and other contaminants. You can find a lot of resources on how this process works by performing a Google search for how do Ampere beads work. The benefit of using ampere beads is that you retain more sample than in a gel purification method. The use of these beads are also automation compatible. So how does this work? Our sample will contain double-stranded DNA, enzymes, adapters, primers, etc., whatever we put into that reaction. We will add beads which will bind to double-stranded DNA based on bead to sample volume ratio, and we can retain specific sizes. Then we will put our tube up to a magnet, and the beads will collect near the magnet along with the DNA bound to it. Enzymes and everything else is left behind in the solution. We will remove the supernatant, do the ethanol wash, elute off the double-stranded DNA from the bead, leaving the beads stuck to the magnet, and transferring the sample to the next step. This is what ampere beads look like. They tend to fall because they are beads and they're heavy. So we always want to keep vortexing to mix them and keep them in solution. This is our recommended magnet stand. Keep in mind that not all magnets are the same. Different magnets can have different strengths. And this magnet is the one we've tested and we know works well with our kits. Here we have an example of what the beads will look like during the workflow. In well 1, we have no beads. You can see here it is clear. In well 2, the beads are mixed with the solution in suspension. We can see that it is a brown slurry. In well 3, we see the beads beginning to congregate near the magnet. So the solution, it still has beads as seen by the brown color, but it is beginning to create a pellet as the beads are beginning to collect. In well 4, the beads have completely bound to the side of the well and it is completely clear. And this is what we want to see before we remove solution and proceed with the wash. We do have a video entitled TruSeq Sample Purification Bead Size Selection and Best Practices within our online sequencing courses, and I have provided the link here for you to access it. So best practices for magnetic beads. Beads need to be at room temperature, so pull them out before using, have them nice and warm. Using cold beads may affect bead performance. The beads are in a viscous solution and they are also very heavy, so they tend to settle. And because of these factors, you will need to vortex the beads thoroughly to make sure that they are always in solution. Use the recommended plate and magnetic stand. The part numbers for these materials are in the user guide. Now, I know that the magnets in particular can be quite costly, and it's tempting to use alternate parts. Keep in mind, as I mentioned, different magnets have different strengths, and we have seen significant differences when using non-recommended equipment. Carefully pipette up and down to minimize sample loss. Do not introduce bubbles, as this can result in the loss of yield. Use freshly prepared 80% ethanol. 80% ethanol that is stale can decrease the percent of ethanol and absorb water from the air, so we don't want that. Do not disturb the pellet with the pipette during elution. The idea here is to flood the well and let it reach the beads. When you remove the ethanol, let it dry a little, as residual ethanol can affect downstream steps. And since we are using the ratio of beads to sample, we can see how aspirating a small amount of extra beads or having beads on the outside of that pipette tip can change the ratio and thus select for an unintended library size, resulting in unexpected shifts and peaks in our trace. So we want to be as accurate as possible with that. In terms of PCR equipment, what we want to highlight here is the importance of using a heated lid. The image on the left is a microsealed PCR performed with a heated lid. On the right, we have the same reaction without a heated lid. On the right, we see condensation on the top. We want to reduce this as much as possible, as it can result in sample loss. We want to heat the thermocycler lid to 100 degrees during incubation to prevent reagent and sample loss. We recommend calibrating the thermocycler to ensure accurate temperatures are being attained. Use the recommended adhesive seals to prevent sample loss due to inappropriate sealing at the edges. This is quite important. Once we have our library made, we want to QC before putting it on the instrument for sequencing. We recommend to check the quality of your libraries with a bioanalyzer. With this method, we can check for the presence of adapter dimer or primer dimer. If we have high yield, it is possible to overload the chip, resulting in high peaks that don't quite make sense. In such a case, we would dilute and reload our sample with an appropriate range. For more information on the different trace phenotypes, again, I recommend registering for that library QC and troubleshooting with the bioanalyzer webinar. For quantification, we recommend to use qPCR. This is the best method to avoid unexpected cluster density, as it amplifies clusterable libraries only that can go onto the instrument. You may ask, why not use qubit? Qubit measures all double-stranded DNA in a sample. So using this method may measure something that won't cluster on the instrument. Additionally, qubit will not pick up bubble product, which can lead to an underestimation of our library and ultimately possible overclustering when you're sequencing. So for more information on bubble product, you can also join that library QC and troubleshooting with the bioanalyzer webinar. Unlike qubit, qPCR will only measure competent libraries. Nanodrop is not recommended as, one, it is not consistent, and two, it takes into account everything, including small nucleotides that are in the sample. Therefore, we do not recommend using it for quantification. We can also talk about general best practices. Properly store the reagents. Reagents within our kit and even within the same box may have different storage temperatures. So pay attention to their labels and store accordingly. Avoid multiple freeze thaws of master mixes by aliquoting reagents. Multiple freeze thaws can affect reagent efficiency, particularly that of enzymes. Carefully handle viscous reagents such as ATL, that is A-tailing mix, and LIG, ligation mix, ensuring to pull the correct amount and without introducing bubbles, as this can affect the amount aspirated and transferred. Adding reagents in the order listed in the user guide is very important. For example, in our TruSeq Stranded Total RNA protocol, we recommend to add our RRB, that's the rRNA removal bead, to the RNA. We recommend doing it in this particular order because if we do it in reverse, we see poor depletion. So the order is very important. Heat and activate at 70 degrees for 5 minutes after A-tailing. Do not over-amplify during your PCR. There is a balance between the number of PCR cycles for sufficient yield and increasing yield. And the recommendations that have been tested that we give happen to be that nice sweet spot. Now, so far we have discussed general best practices, which can typically be used with the TruSeq RNA v2, our stranded mRNA, and our stranded total RNA workflows. Now let's take a minute to discuss best practices for the Illumina RNA Prep with enrichment workflow, which, as you know, is a little different from the others in that it is enrichment-based. So we will make our libraries and then enrich for the libraries of interest. In regards to this workflow, we want to ensure that the beads used, such as the EBLTL and Ampere XP, are brought up to room temperature. As mentioned for other bead types, cold beads do not work as efficiently, and they should never be frozen. We want to ensure that we fully resuspend the beads to ensure that we are aspirating a similar concentration of beads each time for each well. Prepare that fresh 80% ethanol. As always, do not allow the beads to dry. The index adapter plate wells are single use only for this workflow. And keep in mind that we do want to select the appropriate number of cycles for the PCR program, as in this workflow, the number of cycles used will vary based on the RNA input quality that we used. When it comes to pooling and hybridization, we want to use either 1 or 3-plex for enrichment. If using the RVOP panel, that's the respiratory virus oligo panel, we want to use a 1-plex enrichment for this workflow. Preheat the microheating system to 50 degrees. Fully resuspend and preheat the NHB buffer for 5 minutes, keeping it warm to prevent any precipitates from forming. And add the hybridization reaction reagents in the order listed in the protocol. If these steps are not performed properly, it can result in inefficient hybridization and thus a final library pool which is not enriched for the targets of interest. In regards to the capture and wash step, we want to ensure use of the correct beads. In this case, we are using the SMB beads. Use bead handling best practices. Preheat the microheating system and the EEW reagent. Ensure that the thermal cycler runs continuously throughout the capture and washes and prepare throughout the washes and the capture, and prepare a fresh elution master mix. And this will be done with that HP3. These steps will ensure that we are enriching, or in other words, capturing our targeted libraries. Now finally, we want to adhere to the best practices for bead cleanups, ensuring that the correct cleanup beads are used at the right conditions. I would like to quickly touch upon some resources that you may find helpful when creating your RNA-Seq libraries. And these are our support pages, our webinars and bulletins. Our support pages contain the FAQs, user guides, and other useful information. Our webinars you can definitely catch up, such as with part 1, and our bulletins also contain a lot of useful information in bite-sized quantities. Here I've listed support pages, which again contain those best practices, FAQs, and even links to trainings. I recommend visiting them before the start of your project and definitely before the start of your prep. To reiterate, today is part 2 of the RNA-Seq webinar series. If you missed part 1, you can still access it as a recorded webinar. The link is provided here. And I encourage you to register for part 3, so keep an eye out for that. You can also register for the library QC and troubleshooting with the bioanalyzer webinar. The link to register for this webinar is provided here. And I also encourage you to check out our complete list of recorded webinars, as they not solely focus on library prep, but they include instrumentation and analysis topics as well. We also have many useful support bulletins available on our website. Here I have highlighted a few which are useful specifically for RNA sequencing. So I suggest taking a look at them. And with that, I am happy to take questions.